Hi, everyone. We are going to talk about the biology of infectious diseases today. All right. Section 8.1 is just some basics. So microbes is a general term. When we say the word microbe, we're talking about microscopic organisms such as bacteria, viruses, protists. They're widely distributed in the environment or inanimate objects and on living things. They live in every single place you could think of. Many are useful to humans. They contribute to the production of yogurt, cheese, beer, bread, wine, and many pickled foods. Drugs, meaning prescription drugs, are produced by bacteria. Without the activity of decomposers, the biosphere would not exist. It would not function properly if we couldn't break down dead matter and incorporate it back into the soil. Pathogens are a specific microbe that cause diseases. Bacteria, viruses, parasites, fungi, and prions are our main categories of pathogens. We do have defenses against pathogens. We have barriers to prevent pathogens from entering the body, the skin, the mucous membranes. Phagocytic white blood cells fight infection after a pathogen gets past those barriers and into the body. We also have acquired defenses that kill pathogens and protect against cancer. Those we're going to go into when we talk about the immune system, but there is a general overview of what our bodies do for us to fight pathogens. Bacteria is our first category. These are single-celled prokaryotic organisms. Once upon a time, we talked about the difference between prokaryotic organisms and eukaryotic organisms. Prokaryotes are simpler. They don't have an actual nucleus where the DNA is separated off from the rest of the cell. Their DNA is just kind of all willy-nilly in the center portion, the nucleoid region. And they have a few more organelles. They have ribosomes, they have a cell membrane and a cell wall. There are three common shapes of bacteria that we look at, that you will look at when you get to especially microbiology class. Coccus is sphere-shaped, Bacillus is rod-shaped, and Spirillum is curved or sometimes spiral-shaped. Most of them have a cell wall in addition to the plasma membrane that contains peptidoglycan. Some antibiotics, such as penicillin, interfere with the production of that cell wall, and some are surrounded by a capsule that allow bacteria to stick to surfaces and protects them. So this is a bacillus, it's a rod shape. I'm not sure why we didn't show you the other two. Cocci is the circle shapes, and then spiral is spiral. Gram staining. Bacteria are classified by differences in their cell walls, which are detected using a process called a gram stain. When you take microbiology, you will be so tired of gram staining. It'll feel like that is all you do in microbiology class, but it is fascinating. Cell walls that have a thick layer of peptidoglycan stain purple and are called gram-positive bacteria. If they do not have the thick peptidoglycan layer, the cells stain pink and are considered gram-negative. Gram-negative bacteria have an outer membrane with lipopolysaccharides, which are released when cells are killed by the immune system, which will stimulate inflammation and fever. On the left, we have gram-positive. They're purple. They are cocci. They're tiny little circles, but you have a very thick layer of peptidoglycan. On the right, you have gram-negative, mostly rods. They still have a thinner layer of peptidoglycan, but they have the outer membrane and sometimes even a capsule. But they have some extra layers to them. Motile bacteria usually have long, very thin appendages called flagella. Some bacterium have fimbriae, which are stiff fibers that adhere them to the surfaces, such as host cells. Fimbriae allow a bacterium to gain access to the body. Apillis is an elongated, hollow appendage used to transfer DNA from one cell to another. Genes that give bacteria resistance to antibiotics can be passed on through apillis by conjugation. So there's some bacterial structures. On the top left are the different flagella. You can have a lot of flagella or just a few, or even just one on each side. Motility tests, you take a needle and you stab it into agar, And if it stays in the stab mark, that means it's not motile. It doesn't move. It doesn't have any appendages to be able to swim away. If the bacteria is motile and does have a flagellum or fimbriae, it will make that little zigzaggy pattern, meaning they're trying to swim away in the agar. Again, something you will be doing when you get to microbiology. So bacterial DNA. DNA is packaged in a chromosome in the center of a cell. Many bacteria also have small circular pieces of DNA called plasmids. Genes that give bacteria resistance to antibiotics are often located in the plasmid. Abuse of antibiotics increases the number of resistant bacterial strains that are difficult to kill. And that little GIF that is happening over and over again is just an example of antibiotic resistance. You see the pink bacteria come in, and then there's the antibiotics. The antibiotics will kill the pink bacteria, but there is a mutant. That red bacteria is resistant. And that resistance, whatever gene it is, can be transferred to other bacteria. Binary fission is a reproductive strategy that most bacteria go through. The single circular chromosome attached to the plasma membrane is copied. Then the chromosomes are separated as the cell enlarges. In this, the cell just pulls away and separates into two. Bacteria that reproduce by binary fission can reproduce rapidly under favorable conditions, and some can double their numbers every 20 minutes. Strep throat, tuberculosis, gangrene, gonorrhea, and syphilis are well-known bacterial diseases that undergo binary fission. Not only does growth of bacteria cause disease, but some bacteria will also release toxins that will inhibit cellular metabolism. For example, Clostridium tetani produces a toxin that prevents the relaxation of muscles. In time, all the muscles contract, which will cause suffocation. We have harnessed this Clostridium, well, not this exact strain, Clostridium botulinum, to get rid of our wrinkles, to stop the muscles from contracting and giving us wrinkles for a time. People do that on purpose now. Viruses are our next category. They are very small intracellular parasites. They are strictly considered non-living things, acellular, they're not technically a cell, because they must reproduce inside of a host cell. It cannot live independently of a host. It will commandeer the metabolic machinery of a host. There are two parts to most viruses, an outer protein coat called a capsid, and then some kind of a nucleic acid, whether it's RNA or DNA inside. And you can see the relative smallness of it compared to a red blood cell, and then an E. coli is a bacterium, and then they get smaller and smaller and smaller. Portion of the virus bind to a receptor on the host cell's surface. Once the virus attaches, its DNA or RNA will enter the cell and code for the proteins in the capsid. The virus may have genes that code for enzymes needed for the virus to reproduce and exit the host cell. A virus relies on the host's enzymes and ribosomes for its own production. The life cycle stages of a virus. There's many variations of this cycle. It depends on the type of virus, but most viruses will attach. The capsid of the virus will combine with a receptor on the surface of a host cell, and then it penetrates. A viral enzyme digests away part of the cell wall, and the viral genetic material is injected into the bacterial cell, or whatever the host cell is. Other viruses will enter the cell by endocytosis. With other viruses, an entire virus might enter the host cell by endocytosis, and not just the genetic material. The third step is called biosynthesis. The host cell's machinery replicates the virus's genetic material, produces multiple copies of the capsid protein subunits. Maturation or assembly. The viral genetic material and capsids combine, producing several new viruses, and then release. New viruses leave the host cell via exocytosis or by disrupting cell membranes and cell walls in bacteria, which will just release the new viruses out into the open or the bloodstream, wherever it may be. So the GIF is kind of a general 3, 4, and 5, there's biosynthesis. Host cells machinery is replicating the genetic material. It matures and then it bursts out. And then there is the lysogenic cycle. Infected cell does not immediately produce new viruses. The virus will become latent, some viruses, where it is not actively reproducing upon entering the host cell. Following attachment and penetration, integration will occur. Viral DNA is incorporated into the host cell. That becomes what's called a prophage. DNA is replicated along with host DNA. All subsequent cells, the lysogenic cells now, carry a copy of the viral information. Certain environmental factors like UV light, ultraviolet radiation can induce the prophage to enter the lytic stage of biosynthesis, followed by maturation and release. Prions are infectious protein particles. They cause degenerative diseases of the nervous system. Creutzfeldt-Jakob disease, or CJD, in humans, scrapie in sheep, and bovine spongiform encephalopathy in cattle, commonly called mad cow disease. These are all caused by prions. Normal forms of proteins fold into abnormal shapes called prions, and prions are able to change the shape of other proteins. Diseases and human health. An infectious disease is classified as an epidemic if there are more cases than expected in a certain area for a certain period of time. We all have experience with epidemics these days. A few cases of a very rare disease may constitute an epidemic, whereas a larger number of a very common disease may not. An outbreak, an epidemic confined to a local area. So the 2013-2014 Ebola outbreak in West Africa is just considered an outbreak because it stayed in that one area. Global epidemics are called pandemics. We just went through one a few years ago, but those have all had their day as a pandemic. HIV, COVID, tuberculosis, malaria, flu, certain flus, they're all current. AIDS, HIV/AIDS is one that we want to talk about, that everybody should know about. The further away we get from the 80s, the more we forget about the importance of HIV and AIDS. So the exact dates of the first human case of HIV are still being investigated. The first human infection was between 1884 to 1924, so it's actually an older disease. HIV was found in human tissue and blood samples taken in the 1950s and 60s. And the name AIDS was coined in 1982. HIV was found to be the cause of AIDS in 1983 to 1984. HIV structure consists of 2 single strands of RNA, which is its genome, various proteins, and envelope, but it has to acquire the envelope from its host cell. RNA is protected by 3 protein coats, the nucleocapsid, the capsid, and the matrix. Within the matrix are 3 enzymes. Reverse transcriptase converts viral RNA into viral DNA, called reverse transcriptase because it goes backwards to how our body normally does it. Integrase integrates the viral DNA into the DNA of the host cell, and protease converts the newly synthesized viral polypeptides into functional viral proteins. GP120S is a protein spike embedded in HIV's envelope. It must be present for HIV to enter its target cells. A retrovirus is a virus with RNA instead of DNA that must use the reverse transcription to convert its RNA into DNA before it can insert its genome into the host cell's DNA. HIV life cycle. It goes through attachment. HIV binds to a target cell. GP120 binds to a CD4 receptor on the surface of a helper T cell or a macrophage. Fusion. HIV fuses with the plasma membrane and enters the cell, just like is happening in the picture. And then entry. During a process called uncoating, the capsid and protein coats are removed, which releases the RNA and viral proteins into the cytoplasm of the host cell. So hopefully you can see all of that happening in the GIF as I talk about it. Number 4, reverse transcription. This is the process unique to retroviruses. Reverse transcriptase, which is the enzyme, will convert HIV's single-stranded RNA into double-stranded viral DNA. And then integration, the viral enzyme integrase will integrate the new viral DNA into the host cell's DNA. HIV is now called a provirus because it is a part of the cell's genetic material. Again, all of what I'm talking about is happening in those pictures. Integration continued. HIV is usually transmitted by cells with proviruses. Even if drug therapy results in an undetectable viral load, there are still proviruses inside of helper T cells. Biosynthesis and cleavage. Normal cell machinery directs the production of more viral RNA. Now that it is in the genome of that host cell, it will just be replicated along with the host cell's DNA. RNA codes for new viral particles and polypeptides that have to be cut up into smaller pieces. That's what the cleavage part is. And it is catalyzed, this whole process is catalyzed by the HIV protease enzyme. Assembly, capsid proteins, viral enzymes, and RNA are assembled into new viral particles. And then budding, the new virus leaves the cell. The new envelope is actually part of the host cell's plasma membrane. So it's our own cells with our own plasma membrane. We don't destroy them because we think they belong to us. The life cycle of an HIV virus includes transmission to a new host. Body secretions contain proviruses inside CD4 T cells. When this fluid comes into contact with the mouth, vagina, or anus, for example, infected cells migrate through its lining and enter the body of a new host. CD4 macrophages are the first infected when proviruses enter the body. When these macrophages move to the lymph nodes, HIV begins to infect more CD4 cells.
Claire, that just want to remind you, we're starting our clinical staff meeting. Please support the family engagement. Okay.
CD4 macrophages are the first infected when proviruses enter the body. When these macrophages move to the lymph nodes, HIV begins to infect CD4 T cells. HIV can hide out in local lymph nodes for some time, but eventually the lymph nodes degenerate, and large numbers of HIV particles then enter the bloodstream. There are HIV vaccines in development. There's a preventative vaccine, which is not a cure, but would prevent infection. There's a therapeutic vaccine, could slow the progression of the disease in a future infection. Scientists have studied more than 50 different preventative vaccines and over 30 therapeutic vaccines. And I will link this. You can always look it up to investigate the further status, the updated status of HIV vaccines. Tuberculosis used to be called consumption because it seemed to consume the patients from the inside until they wasted away. A third of the world's population has been exposed to TB. It's caused by a species of rod-shaped bacterium called Mycobacterium tuberculosis. It's spread by droplets introduced into the air when an infected person coughs, sings, or sneezes. Sound familiar? It can float in the air for hours and still be infectious. An incubation period is 4 to 12 weeks. It will develop very, very slowly. So 3 months ago, you might have been coughed on by someone with tuberculosis and you would never know it. In the lungs, it is consumed by macrophages. Other white blood cells rush to the infected area. Together, they wall off the original infection site, producing small, hard nodules or tubercles in the lungs, hence its name. The bacteria remain alive within the tubercles, but the disease does not progress. These patients are said to have latent TB. They do not feel sick and are not contagious, but they will test positively on a TB skin test. Tubercles also often calcify and can be seen on a chest X-ray. In a person with active TB, the tubercle liquefies and forms a cavity. The bacteria can then spread from these cavities throughout the body, and it can be fatal. Symptoms of active TB include a bad cough, chest pain, coughing up blood, or sputum. As the disease progresses, symptoms include fatigue, loss of appetite, chills, fevers, and night sweats. The patient begins to lose weight and wastes away. Due to the resurgence of antibiotic-resistant strains, multiple anti-TB drugs are given simultaneously for 12 to 24 months. That's how long it takes to get rid of an active TB outbreak. There are several drug-resistant forms of TB. There's multi-drug resistant and extensively drug resistant. It takes at least 6 months to kill all the tuberculosis bacteria in the body. And then we go overboard just in case. Malaria. So to understand malaria, we got to talk about vectors. A vector is a living organism, usually an insect or an animal, that transfers the pathogen from one host to another. In this case, it's the mosquito. It's pretty common knowledge that that mosquitoes carry malaria. The parasites that cause malaria belong to the genus Plasmodium, which are protists. There are 4 species that infect humans, and the geographic distribution of malaria is explained by the mosquito vector that depends on temperature and rainfall. Mosquitoes don't like cold places, so they're not going to live there. They're not going to spread malaria very much in the cold places. So here's just a general outlook. Female Anopheles mosquitoes spread the parasite in humans by biting an infected person first, and then carries the parasite from that infected person to another. Once inside the new host, the parasite flows through the bloodstream to the liver, where it will multiply rapidly. The parasite destroys red blood cells, causing flu-like symptoms that include high fever and muscle aches. If not treated, it can lead to kidney failure, seizures, coma, and death. Malaria is a life-threatening disease that affects nearly 212 million people worldwide. Anti-malarial drugs will kill the infection. There's also something called the sterile insect technique that scientists are trying to go with right now. It's a form of insect birth control using irradiation. The process involves breeding and segregating billions of male mosquitoes in dedicated facilities, sterilizing them with X-rays, and then releasing them to mate with the females in the wild. The females don't produce many offspring, the insect population will decline over time. And then on the right, there's some ideas on how to protect yourself from mosquito bites. Some people are more prone to mosquito bites than others. We say that they like sweet blood in my family, but empty water near your home, any stagnant water, can be a breeding ground for mosquitoes. Make sure you have no standing water, especially in the hot summers. Use window screens or nets, use mosquito repellent, wear light-colored clothing, especially outdoors. Stay indoors during dusk and dawn. That's the time they like. And make your aroma less appealing. They like sweet smells. Influenza, which is the full name of the flu in case you didn't know, affects 5 to 20% of Americans each year and causes an estimated 36,000 fatalities. It is a viral infection that causes a runny nose, cough, chills, fever, head and body aches, and nausea. You can catch it by inhaling virus-laden droplets or by contact with contaminated objects. Hopefully, most of us have learned to stay away from contaminated objects these past few years. And also cover your mouth when you cough and you sneeze. Use hand sanitizer. The virus is then attached to and infect cells of the respiratory tract. And some emerging diseases and COVID-19. Emerging diseases occur for the first time in humans, are rapidly becoming more common in humans, or are entering into new geographic regions. The avian flu, the swine flu, Middle East respiratory syndrome, MERS, Zika, or SARS-CoV-2. Re-emerging diseases reappear after a significant decline in incidence. So measles and TB are 2 that happen to be re-emerging. Coronaviruses are named for their crown-like spikes emerging from the virus's surface. It is a common animal virus, which we call a zoonotic virus officially, that may infect various animal species, not just humans, but bats, camels, birds, livestock. First human infections were in the 1950s, associated with mild cases of the common cold. Coronaviruses may cause respiratory and intestinal diseases in livestock. In humans, it's the SARS/MERS class of viruses that cause diseases. MERS, again, stands for Middle East Respiratory Syndrome, a zoonotic virus, meaning it's transmitted between animals and people. MERS-CoV has been identified and linked to human infections in dromedary camels in several member states in the Middle East, Africa, and South Asia. Human-to-human transmission is possible and has occurred predominantly among close contacts and in healthcare settings. Outside the healthcare setting, there has been limited human-to-human transmission. It causes pneumonia and kidney failure and has a 34.4 mortality rate, which is actually really, really high. SARS stands for Severe Acute Respiratory Syndrome. It is a more severe form of human coronavirus with a fatality rate just under 10%. It is airborne, transmitted by breathing, coughing, sneezing, or contact with infected surfaces. Everybody should know that by now. SARS-CoV-1 was thought to have originated in bats or civet cats, then transmitted to humans. It appeared in China in 2002 and then spread, causing a global outbreak in 2003, with no further human cases reported since 2004. But over 8,000 known cases and 774 deaths worldwide has been caused by that first version, SARS-CoV-1. SARS-CoV-2, originally called 2019 nCoV and novel coronavirus 2019, is the new coronavirus that causes the respiratory disease COVID-19. It emerged in the Wuhan province of China. It is highly contagious. It quickly became a global pandemic. Hundreds of millions were infected, ever increasing death toll. It caused a global disruption of society by impacting travel, economies, and social interactions. Antibiotic resistance is also a huge problem right now. Antibiotic use does not cause humans to become resistant to the drugs. The pathogens are the things that become resistant. In any given population of pathogens, there are some organisms that are naturally resistant to the drug. They have acquired this resistance through mutations or interactions with other organisms. The drug kills susceptible ones, but leaves naturally resistant ones to multiply. And the new population is then resistant to the drug. That is just the simplest explanation for antibiotic resistance. So development, here comes the drug. It kills most of the bacteria, but it leaves one that happens to be resistant. And that one that survives multiplies, and that new population is now all resistant to whatever that first drug was in the first place. Tuberculosis, malaria, gonorrhea, Staphylococcus aureus, and Enterococci, or group D streptococcus, are a few of the organisms that have developed antibiotic resistance. More and more organisms are becoming multi-drug resistant, leaving healthcare facilities with few choices for treating infections. Prevention of antibiotic resistance. It is very important to take all antibiotics as prescribed, as directed. Do not skip doses or discontinue treatment when you feel better. You have to take it all, no matter how you are feeling. Do not take antibiotics for viral infections, such as colds or the flu. Antibiotics are specifically for bacteria. Do not save unused antibiotics or take them prescribed for a different infection. Each year in the United States, at least 2.8 million people become infected with antibiotic-resistant bacteria, which results in 35,000 deaths. Multi-drug resistant organisms, MDRO, is the abbreviation for that if you ever hear it. There are extensively drug-resistant tuberculosis, resistant to almost all the drugs used to treat TB, including the first-line antibiotics, which are the older and cheaper ones, as well as the second-line antibiotics, newer and more expensive ones. MRSA stands for methicillin-resistant Staphylococcus aureus. It is resistant to methicillin and other antibiotics related to it. It is the cause of quote-unquote staph infections. In 1974, only 2% of staph infections were caused by MRSA, but by 2004, which was 20 years ago, the number of cases had risen to 63%. Who knows what it is now? Vancomycin-resistant Staphylococcus aureus, or VRSA, is not found yet in the US. But vancomycin is literally the strongest antibiotic that humans use, and there are strains of bacteria that have become resistant to it. So fortunately, it's not found here yet, but MRSA and VRSA are mostly transmitted in healthcare settings, in hospitals, in clinics. And that is all for Chapter 8.